Previously, we talked about how the genes of DNA code for the production of proteins. So it's very important that we have to turn certain genes on when we need to make proteins. But we don't need to be making proteins all the time and in every cell. So therefore, we need to regulate that, and that's what we're going to talk about in Chapter 16, entitled Gene Expression. So each somatic cell has a full genome of the organism. That means it has every chromosome and every gene that is needed to make all the proteins and all the materials of for the entire being. But not every cell needs every one of those. So not all genes need to be expressed at the same time in each cell, and that would actually be a bad thing because that would overwhelm the cell and not allow it to do its individual job. So regulation helps maintain efficiency in living beings. It helps preserve energy because if you had to express all the genes all the time, then that would take a whole lot of ATP. It also helps saving space, and it saves time because not all the genes have to be read at the same time. Now, the reading of genes and the making of proteins are a little bit different in prokaryotic organisms than they are for eukaryotic organisms, as we talked about before. Here we have a bacterial cell with its single circular DNA. And when this is transcribed to make its messenger RNA, this blue ribbon, we see that because there's no barrier of a nucleus, that messenger RNA can then enter right into some ribosomes, and the ribosomes can start reading it and converting that messenger RNA into the needed proteins. So how do we regulate this process? Well, because it's all happening at the same time in the cytoplasm, we're going to regulate that right at the transcriptional level, and we're only going to make the messenger RNAs that we need. Whereas in eukaryotic organisms, we find that transcription and RNA processing happens inside of a nucleus. So here's our DNA, the purple ribbon. From that, we make a beginning messenger RNA, but we have to process that messenger RNA and take out the unneeded bits and make changes. And then we have to transfer this messenger RNA out into the cytoplasm so that it can connect with ribosomes and then actually start translating the code into proteins. So we see that these take place in different compartments. Therefore, the regulation can happen during transcription as well as translation, and actually even after translation by modifying the constructed proteins. Here's a great chart which compares prokaryotics, bacteria, with eukaryotic organisms at every level and how regulating genes is different between the two groups. So let's go back to our prokaryotes and take a look at how they regulate their genes, keeping in mind that this is only going to happen at the transcriptional level, at the level of making the messenger RNA. So remember, the DNA is located in a region called the nucleoid region, but not in a nucleus. And therefore, it has access to a class of proteins which can affect the reading of the DNA and the production of messenger RNA. And this group of proteins are called operons. So the operons are regulated by three molecule types. There are repressors. They can suppress transcription or turn it off. There are activators, which can increase transcription or turn it on and make it go faster. And then there are inducers, which may either shut off transcription or activate transcription, depending on what the cell needs. So let's take a look at an example of each of these three options, and we'll start with the repressor operon. So this one is particularly called the TRP, or the TRP operon, for tryptophan. Tryptophan is an amino acid. So when tryptophan is plentiful in the cell and the cell does not need to make new tryptophan, then the plentiful tryptophan will bind to a protein repressor, and this complex will bind to the operator region of the DNA, and this means RNA polymerase can't lock on, and so therefore can't start reading. And therefore we don't make more tryptophan, which we don't need. But if tryptophan is low or absent, then there's no tryptophan to bind onto this repressor molecule. This repressor molecule then will not bind to the operator, and therefore RNA polymerase is allowed to bind to its promoter and continue down this DNA pathway where it will make a messenger RNA that will make tryptophan molecules. Here's an example of an activator protein, one that will increase the reading of the DNA by RNA polymerase. So this is called a catabolite activator protein, or a CAP protein. So these ones in this case are going to impact the reading of lac genes. Lac genes result in the production of lactose sugars. Now in these bacteria, they would prefer to use glucose as their primary sugar. But if their glucose levels are low, then they might have to use other sugars. And so the use of those other sugars might require the production of certain enzymes, and this can be done by activating this system. So if glucose supplies are low in the cell, then cyclic AMP levels increase. The cyclic AMP causes the CAP to bind to its promoter and to the promoter for this region of the DNA, and that will cause RNA polymerase to go really fast down this pathway, making the requirements so that the cell can use lactose sugar instead of glucose. Now, does this happen even when there is glucose? Yes, it will still happen. So even in the absence of cyclic AMP, CAP doesn't bind to the promoter, but the transcription of these genes goes through very slowly. So in this case, we can speed it up, make it go faster when we need to utilize lactose. And then the last one is called the lac operon. So you notice we have the same genes going on here. So in the absence of lactose, so when we don't have any lactose, that means we have plenty of glucose. That's the preferred source, and we don't need to make these components. So therefore, there's a repressor protein that in the absence of lactose will bind here, and so that RNA polymerase cannot hook on, and we cannot go through this pathway. But if we have lactose and the cell can use the lactose, then what happens is the lactose will bind to the repressor and it will dissociate, it will come away from the operator, and then RNA polymerase will be able to bind and will go through and read these genes to make the components so that the cell can use the lactose. So this is one of those either or situations where it depends on what does the cell need. If the cell is low in glucose and needs to use lactose, then it will go down this pathway. All right, now eukaryotes, so everything we've talked about so far has been prokaryotes, and that's only at transcription. But eukaryotes, the gene expression is much more complex than it is in prokaryotes. It can happen, remember, transcription and translation are in two different cell compartments. This is in the nucleus, this is in the cytoplasm. So regulation can happen at different levels. The first level is who gets access to the DNA. That will affect how transcription starts and gets going. The second level is also transcription factors. So this is proteins that control also the starting of the reading of the DNA to make RNA. And thirdly, we're going to look at the RNA slicing. So these are all things that can happen that can affect transcription in eukaryotic cells. So, regulating access to the genes in the chromosome. Remember that the human genome has about 20,000 cells, approximately. And these are located on 23 chromosome pairs. So this means there's thousands of genes on every chromosome. The DNA has to be packed, and so therefore it's wound around these histones. So therefore, some of the genes we might need to utilize could be unable to be accessed because they're wound around histones. So we need to unwind them in order to make them available to the polymerases. And this is just a picture showing the DNA winding around the histones. So these wound up bits of DNA around histones form these nucleosome groups, and you can see, say this is a region, a gene that we need to read, but it's inaccessible because it's kind of tucked into this grouping, and so therefore the polymerase can't get in there. So what we need to be able to do is unwind the DNA, so now the gene is accessible. So when the nucleosomes are spaced far apart, like here, the DNA, we can now read those. When the DNA is exposed, then the transcription factors can bind to it and allow the gene expression to happen. So this is a process of unwinding the histones, and that happens through acetylation in trying to get these histones apart from the DNA. So there's chemical tags, which I alluded to just a bit ago, that are added to the histones and the DNA. So these tags can be phosphates, methyl groups, acetyl groups. These tags are not permanent. They come and go. So they act as signals to tell the histones, do you need to unwind or do you need to wind back up? So this is part of that epigenetic regulation. So remember that certain genes can influence other genes and nuclear proteins in order to affect the expression of the genes. So here we see the unwinding and the opening of the DNA, which is allowing the transcription factors a way to get in to those regions of the DNA that are otherwise inaccessible. So we also have transcription gene regulation. So that was kind of before transcription starts, just getting access to the DNA. How can we affect the actual transcription? So remember that in eukaryotes, we need to have some polymerases, some RNA polymerases, which are going to make transcription factors in order to initiate the transcription process. So in order to make the RNA, we're going to have to give the RNA polymerase the ability to attach to the DNA. And there are special proteins that are going to help that and signal that to happen. And so these factors are going to bind to a part of the DNA called the promoter sequence, and they're going to get things going. So here's a little YouTube video to watch, which will show that. But let's take a look at how that happens. So here we have a piece of DNA that we want to start reading. And we have a promoter. So we have this in front of the gene that we want to read. So in these transcription factors, we talked about those in the last chapter, have to bind to the promoter in that TATA box. That TATA box isn't shown here, but it's right in here. They need to bind in before the RNA polymerase can bind and then get started. Well, additionally, there are these transcription factors can be influenced based on other either activators or inhibitors. So if these other regions have activators associated with them, then this will help the transcription factors bind and get the RNA polymerase going. Here in this particular case, we see that we have a repressor that binds. And so because that repressor binds, that will not allow these transcription factors to let the RNA polymerase in, and we will not get the reading of this gene. Here we will get the reading of this gene. So these are other factors which will affect the transcription factors, not allowing RNA polymerase. Here allowing RNA polymerase to bind, here not allowing RNA polymerase to bind. And the third type was after transcription. So here we are now looking at a piece of RNA, a messenger RNA that has been transcribed. It has 1, 2, 3, 4 coding regions or exons and 1, 2, 3 introns, along with its 5' cap and its 3' tail. Now, it's going to have to go through processing where we take those introns out, but also we may selectively choose only certain regions to be read and turned into proteins. So for example, we could splice out on this side the number 4 region of coding and only have 1, 2, 3. And maybe on another time, based in other sequences, we could have only 1, 3, 4. So we can splice the RNA, the messenger RNA, in different ways after it's transcribed to get a messenger RNA that will result in a different protein. All of this, whether it is opening up the DNA, which was number 1, number 2, limiting access of RNA polymerase to actually transcribing, which was number 2, or taking the transcript and making changes. All 3 of those processes happen in the nucleus. So we also have to be careful of making sure that this messenger RNA remains stable after it has been processed. So we are going to add in a 5' cap, which is methylated guanosine triphosphate molecule. So these are a methylated GTP, and this is going to prevent the degradation or the breakdown of this messenger RNA until we actually get to translate it. And then the poly A tail is also going to prevent breakdown. There are protein coding regions of the messenger RNA. The protein coded regions, the exons, have also untranslated regions on either end. So that's these UTRs, untranslated regions, one on the 5' end, some on the 3' end. These also help to make the RNA molecule more stable so that it can stay intact on its trip out of the nucleus and into the cytoplasm so that it can then begin to be translated into proteins. Now, what happens in the case of cancer is that we can have a situation where gene expression is altered. And if we alter the reading of the genes, we can speed up that reading, and that may affect going through cell cycle and causing the cell to grow more than it normally would. So cancer can result from a gene mutation. So this is some external force like UV radiation or some particular chemical literally changing the DNA. So changing the coding in the DNA, that could result in an altered reading. But we also see that it could be due to problems with gene regulation. So if we affect how fast or how slow genes are read and in what order they're read, and if they're read when they shouldn't be read, then that can cause cells to go into a state of altered being. We're going to look at one specific example of this that results in cancer. Remember a few chapters ago we talked about tumor suppressor genes. So these genes help to prevent inappropriate cell growth. So they hold cells back from going through the cell cycle. And one of these genes is the p53 gene. So it's a tumor suppressor gene. It stops cells from going through their cell cycle and entering into mitosis when they don't need to. And what we found is that this particular gene is mutated in over 50% of all cancer types. So p53, the product of reading this gene, is that it produces a transcription factor. So if we are making transcription factors in excess and we are making too much of them, then that could cause an overexpression of the reading of genes, and we'll no longer be able to inhibit the genes. Additionally, oncogenes and cancer. So remember we talked about proto-oncogenes. So these are genes which when mutated, when the proto-oncogene is mutated, we call it an oncogene, and these will alter transcriptional activity. So when these become mutated and become oncogenes, then they accelerate the reading of genes that cause a cell to go through excessive growth. So one of the genes that gets mutated is the MYC gene, and this codes for a transcription factor that is activated in this particular type of lymphoma. So it's not important to remember these types of cancers, but just know that in cancer, we can either mutate the specific gene or we can mutate a factor, the production of a factor which influences the reading of genes. And so the reading of genes or the expression of genes in the proper timing and the proper amount is what's very key to keeping cells healthy.